how are you? Welcome to our lesson today. Today we shall learn about primary and secondary growth in plants. You will be guided by me, your teacher, Mr. Juma Nyamawi of Wa Girls High School. Welcome and enjoy. Now, have you ever planted a seedling and you take care of that particular seedling, let's say for about 6 months? What events do you normally observe on the seedling? Okay, probably you have said that the height of the seedling increases and also the width or diameter of the stem also increases. So all the two phenomena that you have talked about actually are the two types of growth, that is primary growth and secondary growth. So what is the name given to the increase in height? The increase in height is what is called primary growth. What about increase in girth or diameter? The increase in girth or diameter is what is called secondary growth. Okay? So, what brings about these two types of growth? Primary growth and secondary growth is brought about by presence of some cells in the growing regions. And these cells are called the meristematic cells. We call them the meristematic cells, or simply meristems. So, what are meristematic cells, or what are the meristems? Now, meristematic cells or meristems are actually undifferentiated cells in plants which are capable of continuous mitotic cell division. What does that mean? They are, first of all, cells that are not differentiated, and they can also divide by mitosis continuously. Okay? Now, where do we find meristematic cells? So in flowering plants, meristematic cells are found in the following regions. One, we can find them at the tips of the root and the tips of the shoot. Also, they are found in the young leaves, and they are also found in the internodes, and they are also found in the cambium. Okay, we have two main types of cambium. Yeah, we have what we call the vascular cambium and the cork cambium. So, the meristems that are found at the tips of the root and tips of the shoot are called apical meristems. They are responsible for increase in height, or what we call primary growth. Okay. Also, the meristems which are found at the bases of the internodes are known as the intercalary meristems. So when they divide by mitosis, they lead to increase in size of the internodes, as in monocotyledonous plants like maize and sugarcane. Okay. On the other hand, those meristems which are found at the cambium are of two types. We have the vascular cambium and the cork cambium. They bring about what we call secondary growth or increase in girth or diameter of the stem. Having we learned that, we have said the following, that 1. Meristems which are found at the tip of the shoot are called apical meristems, and they bring about primary growth. On the other hand, cambium meristems are responsible for secondary growth, and intercalary meristems are found at the bases of the internodes. Bases of the internodes hence bring about elongation of the internodes and increase in the sheath of the leaves in grasses and other monocotyledonous plants. Okay, good. Now let us have a look at these particular types of meristems. This is a plant, So at the tip here, we are able to see that at the tip, sorry, okay, yeah, good. So at the tip here, you are able to see that we have said that we have two types of meristems occurring there. We have the apical meristems occurring at the shoot tip and the root tip. Okay, so at the shoot tip, we have some undifferentiated cells here which are capable of dividing by mitosis or mitotically. This is what forms the apical meristems. Also, at the root here, we also have the apical meristems. Okay. Now, if you look keenly at the root tip, or the shoot, sorry, at the shoot tip, we have actually some structures which protects the meristems. These are leaf-like structures produced by the meristems themselves. They usually form a bud, and this bud actually protects the delicate meristems. And this bud is known as, or these structures are known as the leaf primordia. Leaf primordia. If it's one, it's leaf primordium. Okay. Then down here, the apical meristems are found there. Okay, and they are usually protected by the root cap. As the root grows downwards, okay, usually it comes into contact with some tough materials like soil or even rock. So to prevent these delicate meristems from abrasion, from being ground by the soil, we have the root cap. This root cap actually protects what we call the meristems. So these meristems usually divide by mitosis, hence bring about primary growth. Then at the center of the plant here, the stem, we have now what we call the cambium. The cambium actually consists of, we have two main types of cambium. The one which is towards the inner, near the blue. Okay, near the blue. Yeah, the cambium which is towards the inner actually is what is called, this one here is what is called the vascular cambium. So vascular cambium brings about increase in diameter, as we shall see later. Then we have the one which is towards the outer, this one. That one is what is called the cork cambium. Cork cambium is actually important information of the new bark of the tree as expansion of the stem occurs. Okay, let us have a look at the intercalary meristems. So intercalary meristems, on the other hand, are found at the bases of internodes. So this here is the internode. So at the basis of the internode, we have what we call the intercalary meristems. So when these cells found at the intercalary meristems divide by mitosis, it leads to increase in number of cells. This will lead to increase in the internode, as seen in monocotyledonine. Okay. Now, let us have a look at this particular diagram, which clearly shows how the cambium at the stem looks like. Okay, so we have said that the cambium towards the outer is what is called cork cambium. Cork cambium, this one. So the cork cambium is responsible for the formation of new bark in case secondary growth occurs. Then we have the cambium towards the inner. Okay. So this cambium here towards the inner is what is called the vascular cambium. it is hidden down here. Okay, the vascular cambium, okay, the one towards the inner. So the vascular cambium, upon undergoing mitotic cell division, it actually leads to formation of new cells towards the inner, this one which we call the secondary xylem, and two new cells towards the outer, which actually are called the secondary phloem. As the new cells form at the vascular cambium, there will be increase in diameter. The diameter will tend to increase, as we shall see later. Okay, so the cambium usually is responsible for increase in diameter, or what we refer to as secondary growth. Now, what are the characteristics of meristematic cells? Have a look at this particular diagram. What can you see? What are some of the features that are distinct? Do these cells have any cytoplasm? What about their thickness, the thickness of the cell wall? Do they have a subvacuole? So if you have actually considered all those areas, we can say, one, that they have got a very thin cell wall. Okay. Number two, they have dense cytoplasm. You can see almost every part of the cell is cytoplasm. And number three, they lack subvacuole. And if you look keenly here, we have one cell which is dividing on top here. Yeah, one cell is dividing here. One cell is dividing by mitosis. Another cell is starting to divide here. So the cells actually divide mitotically at any given time. Okay, good. So we have said the following, that one, they have got dense cytoplasm, and number two, they have thin cell walls, and number three, they lack vacuoles. So these are the characteristics of meristematic cells. Now, I want us to discuss how primary growth occurs in plants. What brings about primary growth? Now, primary growth occur as a result of events that occur in different parts of the shoot tip. If you look keenly at the diagram displayed here, you'll be able to notice that we have three main zones. Can you identify the zones? We have this zone here. This is what we call zone of cell division. Okay. Then we have this zone here. What is it called? Zone of cell elongation. And finally, we have the last zone here. What is the name of this zone? This is what we call zone of cell differentiation. Now, what are the activities that occur in these three zones? So at the zone of cell elongation, you are able, at the zone of cell division here, you are able to see that at the tip here, we have the apical meristems. So the apical meristems will divide mitotically or by mitosis, hence bring about formation of new cells. So as the cells build up, it will actually bring about increase in size. However, so here we are saying the characteristic is that the cells at this particular region divide by mitosis. Okay. Then the apical meristems, or the cells which are found here, usually give rise to what we call the leaf primordium. The leaf primordium, this one here, is the leaf primordium. So the leaf primordium is important. Why? Because it forms a bud which protects the delicate meristems. Now, what are the events occurring at the zone of cell elongation. So at the zone of cell elongation, usually the cells that have been formed at the apical meristems, okay, leads to they do elongate by forming some vacuoles. So vacuoles start to form at this particular point. Hence, the cells elongate. Then what about the zone of cell differentiation? So at the zone of cell differentiation, there are actually, okay, the cells usually differentiate into permanent tissues. That is, they form tissues. Let's say, for example, these ones here. If you cut the stem at that particular point, you're able to see that we have some permanent tissues, like we have the vascular tissues, the cortex, epidermis, the pith, the vascular bundles, etc. Okay, so these tissues usually are formed at the zone of cell differentiation. Okay, so I've said we have three main zones. We have the zone of cell division, where cells divide by mitosis, or we can say they divide mitotically. Also, we have zone of cell elongation, where cells elongate to maximum size. And then finally, we have zone of cell differentiation, where the cells differentiate to form their tissues, or what we can say, to form their permanent tissues. Okay? Then, which structures protect the apical meristems? We have talked about the leaf primordia. Let us have a look at what happens now at the shoot, at the root. At the root tip also, three zones are visible. We have this zone here. This is what we call zone of cell division. Here, there are also apical meristems as observed. We have the apical meristems here. Okay, so the apical meristems usually divide mitotically. So as they divide mitotically, they bring about new cells. Okay, so when new cells form, it leads to increase in size. The characteristics of cells at that particular point is that, as discussed earlier, one, they have got dense cytoplasm, they lack vacuole, and finally, they have got very thin cell wall. What protects these delicate cells at the apical meristems in the root? We have talked about the root cap. So the root cap is the one which protects the delicate apical meristems. So these apical meristems are found at the zone of cell division. Let us now talk about the zone of cell elongation. As in stem, also at the zone of cell elongation, usually the cells which are formed at the zone of cell division elongate to their maximum size by forming vacuoles and lengthening of their walls. Okay? Therefore, we can say at that particular point, the cells start to form vacuoles. Lastly, we have the zone of cell differentiation. So this is where the cells differentiate into permanent tissues. Like there will be some tissues such as the cortex, which will form, such as the phloem, such as xylem, the vascular cambium, etc. So this particular point, it is where now the cells form permanent tissues. And in roots, it is characterized by formation of a lot of root hair cells. There are some root hair cells which are visible there. So that actually is what actually forms part of the zone of cell differentiation. Okay, good. Now let us go to the literature so that we understand so well about these particular zones. Good. Now we have said that primary growth occurs at the root tip and the shoot tip, and these areas, it is where we have the meristems. We have also said that at the tip we have zone of cell division. This is where meristematic cells divide by mitosis. We have also said that below this region we have zone of cell elongation, where cells elongate to their maximum size by stretching of their walls. And we have said at that particular point, vacuoles start to form. Okay, good. Then the elongated cells will later differentiate into permanent tissues at the zone of cell differentiation. The cells here are large and they have got large vacuoles and thickened cell walls. We have said they differentiate into permanent tissues. At the tip of the stem, we have said we have leaf primordia. If it's one, it's leaf primordium, which actually forms a bud. And the main function of this is to protect the bud, it is to protect the delicate meristematic cells. At the root, we have said the meristems are protected by the root cap. So that's all about primary growth. Hope you have enjoyed it. So let us meet again as we shall be discussing about secondary growth. So have a nice time. Bye bye.